Hello, everyone, and welcome to today's seminar. My name is Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis. The center represents a collaboration between 9 labs across New York City, interested in developing tools and methods focused on multimodal single-cell analysis. This seminar is part of our recently launched virtual series, where we will showcase new papers and methods that stem from our center and that we think that will be interesting for the broader biological community. Before we start, I want to go through a couple of organizational details. Today, we're going to have one talk, and at the end of the talk, there's going to be time for you to ask questions. If you want to ask the question, please raise your hand, and I will call on you, and you will be able to unmute yourself and ask the question. If you rather have me read your question, please type it in the chat box, and I will read it to our speaker today. With that, I'm going to introduce our speaker today, Efthymia Papalexi. Effie is a graduate student, a PhD student at the New York University, and a member of the CITE-seq Lab at the New York Genome Center. Her research is focused on single-cell perturbation screens and how to develop better methods to better interpret them. With that, I'll hand it over to you, Effie.
Hello, everybody. Thanks for the introduction, Sandra. Let me just share my screen. Okay, can everybody see my screen?
Yes, we can.
Thank you. All right. So thank you for joining today. I will talk to you about my recent publication on trying to understand how inhibitory immune checkpoints like PD-L1 are being regulated. Just a little bit of a background. T cells are able to kill pathogens, but they're also able to kill cancer cells. And for that to actually happen, first, you need to have T cells be able to actually identify the pathogens or the cancer cells as something bad for the body. And also, you need to be able to have these T cells sort of activate or change their molecular state in order to be able to secrete toxins and cause the apoptosis of these cells. So how do T cells become activated to kill? Well, T cell activation is a two-step process. The first thing that needs to happen is you're going to have your T cell receptor bind a pathogen or cancer-specific antigen coming from the antigen-presenting cell. And at this point, the T cell is aware that there is something going on around it, but it's not. It hasn't been fully activated yet to act against it. This is where immune checkpoints come in. The immune checkpoints will determine if the cell will proceed with its activation or not. They come in pairs of a receptor and a ligand, with the receptor residing on the T cell side. And immune checkpoints come in two flavors. They can be inhibitory and they can be stimulatory. Each category has multiple immune checkpoints in them. So the balance between the two is going to not only determine whether a cell will be activated or not, but also how strong the activation will be. In other words, immune checkpoints are extremely important because they can sort of modulate the amplitude of the immune response, but they can also, they're also very important because they can sort of ensure that you're not attacking yourself. Just to give you an example of an inhibitory immune checkpoint, the pair of PD-1 receptor and its ligand PD-L1, upon binding of the ligand to the receptor, you get the message across to the T cell that it should not be activated. It turns out PD-L1 is an important inhibitory immune checkpoint ligand because a lot of tumor cells upregulate PD-L1 to be able to escape immune surveillance and thrive. And so there has been a lot of interest in understanding the regulators that of PD-L1 expression in order to find ways, new therapeutic avenues to be able to target in patients with tumors, high PD-L1 expression tumors. So I became really interested in identifying new regulators of PD-L1 expression during my PhD. And just before I go into how I wanted to identify these regulators, I'll just give you an overview of what we already know in terms of PD-L1 regulation. So at the transcriptional level, we know that there is IRF1, the IRF1 transcription factor, and also NRF2, coming from two different types of pathways. They can upregulate PD-L1 transcript expression. At the post-transcriptional level, we have a ubiquitin ligase complex between CUL3 and SPOP that will actually add ubiquitin to the PD-L1 protein and tag it for degradation. And at the epigenetic level, we have, there's some evidence that BRD4, an acetylation mark reader, is a positive regulator of PD-L1 that actually, recruits the necessary transcriptional machinery for PD-L1 transcription to occur. So I wanted to basically add something to this scheme of known regulators. And the first thing that I did is I established an in vitro model system where I took a cell line and I stimulated with different cytokines. For example, I stimulated cells with interferon gamma. And then I checked to see if the stimulation results in an upregulation of PD-L1 surface protein expression using flow cytometry. Once I had an in vitro system that actually gave me this PD-L1 upregulation, I wanted to leverage a single cell technology called CITE-seq to be able to correlate PD-L1 surface protein expression with changes in the transcriptome of the cells, in the hopes that that way I can identify putative regulators of PD-L1. So for those of you who are not familiar with CITE-seq, CITE-seq is a single cell RNA sequencing technology that allows for surface protein measurements from single cells as well. The way we're able to measure surface protein expression with CITE-seq is by taking an antibody and conjugating it to a DNA barcode that has a poly-A tail. So essentially what we're doing is we are mimicking a poly-A tail transcript, so that when we do our single cell RNA sequencing experiment and we have our poly-DT beads to capture all the polyadenylated transcripts, this DNA barcode will also get captured. And in other words, what we're doing is we're taking an antibody and convert it into a sequenceable readout. So when I applied CITE-seq to my in vitro model system, I basically wanted to make sure I can trust the protein measurements I'm getting out of it. And so on the left panel at the top, I'm showing you, flow cytometry measurements of PD-L1 expression in stimulated and unstimulated cells. And you can see that the stimulated cells upregulate PD-L1 protein by looking at the shift towards the right. When I measured PD-L1 expression with CITE-seq this time, I see a similar shift to the right, suggesting that I can see the same surface protein shifts in both technologies. So once I knew that CITE-seq can detect properly PD-L1 expression, I wanted to take my cells, order them based on their PD-L1 protein expression, and then look and see what are the transcriptomic signatures that correlate with PD-L1 levels. So on the right, I'm showing you a heat map where every column is a cell and every row is a gene. And the more yellow, it means the gene is really highly expressed. The more purple, it means the gene is not expressed. And so you can see that stimulated and unstimulated cells have very different transcriptomic signatures. And you can see that cells are ordered based on how highly expressed they are, how highly PD-L1 protein expression is. And you can sort of see that cells with high PD-L1 expression in the stimulated group also upregulate, for example, IRF1 transcript, which I already told you it is a regulator of PD-L1. So we thought, I'm showing you a list of maybe 10 genes here that coincide with PD-L1 upregulation, but now actually I have hundreds of genes that I can look through and identify other putative regulators of PD-L1. Once I have these regulators of PD-L1 or putative regulators identified, I need to find a way to test and validate them all together in a pooled format. So to do that, I basically used an extension of CITE-seq called ECCITE-seg, which combines pooled CRISPR screens with single-cell RNA sequencing and surface protein expression. So the idea was that I can perturb the gene of interest, a putative target regulator of PD-L1, and then see what are the transcriptomic changes that correlate with the perturbation of that gene, and what are, if any, the changes in PD-L1 protein expression. So I designed a screen targeting about 25 genes. I used a Cas9 cell line. I had about 3 to 4 guide RNAs per target gene, so a total of 111 guide RNAs. I induced indel formation, and I stimulated my cells and ran ECCITE-seq. And just to give you a few of other facts, we had 4 independent transductions of the same virus as biological replicates. We included some unstimulated cells just as a negative control in the screen. And overall, we got back maybe 50,000 cells, and 62% of those cells had a unique guide assignment. So now we took these cells that we have guide RNA assignments for, and we used their transcriptome to cluster them, hoping that our cells will cluster in a perturbation-specific manner. But what we actually found is that cells tended to cluster either based on biological replicate or cell cycle phase, or even stress response that I'm not showing you here. The only cluster that seemed to be perturbation specific was this cluster I'm highlighting here with the oval, that seemed to have cells expressing guide RNAs targeting upstream components of the interferon gamma signaling pathway. So this was a bit disappointing because we have 25 perturbations and we only got 1 perturbation specific cluster. And the reason for this is really the fact that we have these confounding sources of variation that are not allowing us to see the perturbation specific effects that we're causing with CRISPR. If you look at, for example, 3 different cells that express the same guide RNA targeting the IRF1 gene, you'll see that these cells end up in extremely different parts of the UMAP. And that is primarily because cells are in different cell cycle phase, or they've come from a different replicate. But we don't know really, ideally we would want these 3 cells to cluster together because of the effect that this IRF1 guide has on the IRF1 gene. Instead, what we see is that each one of these cells clusters together with a lot of non-targeting control cells. And the reason for that, again, is because, for example, this blue IRF1 cell clusters with a lot of non-targeting cells that are in the same cell cycle phase. So we needed an approach to basically remove all these confounding sources of variation, with the hopes that at the end we will be left with only the signature that is responsible, or that is coming from the perturbation we have caused. So we developed a method to calculate the perturbation signature for every cell, which is a very simple and intuitive sort of procedure. So we take a cell in our data set, say we take this blue cell here on the top, and then we ask, what are the nearest non-targeting control neighbors of that cell? So at the bottom, we find the 20 nearest non-targeting control cells. Once we have these non-targeting control cells, we average their expression, and subtracted from that initial IRF1 cell. Now, if the cells are sharing the same cell cycle phase, once we do that subtraction, the gene expression matrix for those cell cycle genes should end up being 0. Yeah, so anything that would not be 0 would be specific to the perturbation that the guide RNA the cell has received, has caused. Okay, so once we calculate the perturbation signature for every cell in the data set, then we can recluster the cells and see if we can if we have removed the confounding sources of variation. And the answer is yes. We see that there's no replicate specific clusters or no cell cycle phase specific clusters. And further, you can see that now, in addition to having these upstream components of the interferon pathway cluster, we also have another perturbation specific cluster, primarily composed of IRF1 guide cells. So this was promising, but still we didn't get quite as many perturbation specific clusters as we hoped. The reason for that is because we have an additional source of variation in the data set that we never considered before when analyzing the cells. So this is what we found. It turns out, if you look at cells that express one single guide RNA targeting the interferon gamma receptor 2 locus, all these cells are in red, you'll see that there is cells that form their own cluster, but there's also cells that sort of overlap together in the same cluster as the non-targeting control cells. And so this made us think that maybe some of these cells, despite the fact that they're expressing a guide RNA, perhaps they are not perturbed. They have escaped somehow CRISPR perturbation. So if we split these red cells into 2 subgroups, the cells we think within the oval are the true knockout cells, and then the remaining red cells as the cells, what we would call them as being non-perturbed. We have a few checks we can make to actually see if our hypothesis here is correct. So one thing we can look at is look at the PD-L1 protein levels. We know from literature that if we knock out interferon gamma receptor 2, cells, PD-L1 expression in those cells should go down. And so we see that in the cells we have identified as the true knockouts, PD-L1 protein levels are much lower than the non-targeting controls. But for the cells we called non-perturbed, you see that they have similar levels to non-targeting control levels. Moreover, if you look at the transcriptome of these cells, you'll see that only the knockout cells have a true downregulation of all the interferon gamma signaling pathway genes. And the non-perturbed cells upregulate these genes the same way the non-targeting cells do. Okay, so why do we think these cells are perturbed or non-perturbed? So to answer that question, we went and we got very lucky to be able to recover sequencing reads that are at the locus where the interferon gamma receptor 2 guide is targeting. If you see, the non-targeting control cells have no indels in that particular target site, but both interferon gamma receptor 2 non-perturbed and knockout cells seem to have insertions and deletions there. But when we quantified the types of insertions and deletions, meaning are these frame shift mutations or other in-frame mutations, we noticed that non-perturbed cells had a lot of in-frame deletions. So there were either 3 base pair or 6 base pair deletions, suggesting that when you remove just one amino acid from the protein, the final protein product, you perhaps don't mess or you don't affect the protein function. So we thought that these non-perturbed cells are masking, the signal we can get from the data, and we needed a way to systematically identify them and remove them from the data set. We basically, if we look at these cells that we know express the guide RNA targeting this gene, we want to be able to separate the sort of cells that have a star, which are true knockouts, to the cells that just express the guide RNA but are not perturbed. Say we had these handwritten numbers, and we all classified them as the number 2. They might all be the number itself, but we could potentially find subcategories of 2s, better 2s, and maybe 2s that look more like 7s, and maybe we want to discard those. Sort of, that's in general the idea. So if we take our cells that express guides targeting interferon gamma receptor 2 gene, and we want to summarize in one number what is the strength of the perturbation they have received, and then order them based on that strength of the perturbation, you get sort of a distribution looking a lot like this red distribution I'm showing you here. And you can kind of see there's 2 peaks, suggesting that maybe we have a mix of 2 distributions, one describing the cells that are non-perturbed, and another that describes the cells that are true knockouts. Now, we developed a method called Mixscape to be able to identify these non-perturbed cells. And it's using a Gaussian mixture models to be able to estimate these 2 distributions and classify the cells of whether they belong to one or the other distribution. Of course, one of the distributions we already know because it should fall on top of the non-targeting control cell distributions, since non-perturbed cells should have a very similar transcriptome to the non-targeting cells. And so if we apply Mixscape and we classify cells as whether they're knockouts or non-perturbed, you see that all the cells we classify as non-perturbed sort of fall exactly on top of the distribution of the non-targeting control cells, which was nice to see. But we have, in the ECCITE-seg screen we have a lot of positive controls, and we have an additional assay, which is the protein, PD-L1 protein expression that we can verify our Mixscape classifications with. So in the screen, we targeted a lot of the upstream components of the interferon gamma pathway, the receptor, JAK2, STAT1, transcription factor, and IRF1. And we know from literature that if we knock out any of these components, we should see a reduction in PD-L1 protein expression. So if we look at the PD-L1 protein expression in cells sort of grouped by the target gene class they belong to and split by whether they're classified as knockout or non-perturbed by Mixscape, we see that only the true knockout cells show a reduction in PD-L1 protein. So that was encouraging to see. And so after we are sure that Mixscape is working properly, we went ahead and applied it to all of the cells in the data set, and actually, we could quantify for every target gene class what is the percentage of knockout and non-perturbed cells. And we can split even further our data set based on which guide RNA a cell received. And you can see that with Mixscape, we can perhaps estimate guide RNA targeting efficiency as well. So here I'm sort of I have a rectangle around these IRF1 guides, and you can see that IRF1 guide 1 and guide 2 have a lot more non-perturbed cells than guides 3 and 4, suggesting that perhaps guides 1 and 2 have a low targeting efficiency. And we went ahead and validated this. At the bottom panel, you see the flow cytometry results when we did single guide RNA knockouts and looked for shifts in PD-L1 expression. You see that IRF1 guide 4 and guide 3 are the ones with the strongest downregulation of PD-L1. And so you can kind of appreciate that if we just rely on looking at flow cytometry results and single guide RNA knockouts, we might miss out the true signal if the guide RNA is not efficient enough at targeting. And so by removing the non-perturbed cells from a data set, we can actually boost our signal to discovering new regulators. Okay, so overall, out of the 25 genes we perturbed, now after removing all these non-perturbed cells, you can see that we get a lot more perturbation-specific clusters for other genes that previously we couldn't identify. And you can see that each one of these perturbations comes with a unique transcriptomic response that doesn't necessarily overlap with all the other perturbations. We come a full circle here to say we started with the goal of identifying regulators of PD-L1 expression. So now that we have the knockout cells, what can we learn? We are able to find 8 regulators of PD-L1 in our screen. The upstream path, the interferon gamma pathway upstream elements we already knew about. And then we also identify some negative regulators of PD-L1, BRD4, MYC, and CUL3. And I'll focus a little bit on CUL3 just to say that in the beginning, I told you CUL3 is a post-transcriptional regulator tagging PD-L1 protein for degradation. But when we looked at our CUL3 knockout cells, we found that not only they upregulate PD-L1 protein, but they also had higher levels of PD-L1 transcript. And so this was surprising, and we further investigated it, and we asked what are the genes that are essentially being upregulated specifically in the CUL3 knockout cells as a way to figure out, how do we get elevated PD-L1 transcript levels. And what we found is that all these genes that are upregulated in CUL3 knockout cells are part of the NRF2 pathway. And so what we thought is, just by looking at literature, we found that CUL3, together with a different protein this time, KEAP1, forms a complex and tags NRF2 protein for degradation. So essentially, when we knock out CUL3 in our system, we not only allow for PD-L1 protein levels to accumulate in the system, but we're also allowing for NRF2 protein to accumulate and perhaps go to PD-L1 promoter and further induce its transcription. And we validated sort of this involvement of NRF2 and KEAP1 in PD-L1 regulation by doing a couple of things. First, we did a small pooled CRISPR screen with targeting CUL3, KEAP1, SPOP, NRF2. And then we showed that when we look at the PD-L1 high expressing cells, we see that they're enriched with guides targeting KEAP1, CUL3, and SPOP. And conversely, they are depleted from NRF2 guides and also our negative control, which was interferon gamma receptor 1 guides. Further, we also overexpressed NRF2 in our cells to show that we have an upregulation of both mRNA and protein, PD-L1 mRNA and protein levels, and also did single guide KEAP1 knockouts to show the same effect. So overall, I hope to sort of have convinced you that when we are able to calculate the perturbation signature in our data set, we can remove confounding sources of variation. And the important thing here is that we can do that without even knowing what these sources of variation actually are to begin with. You can I've already told you that sort of some of the CRISPR escaping cells that we see in the data set are coming from in-frame deletions. With Mixscape, we can identify the cells and remove them from the data set, increasing our signal to noise ratio. One of the things we found by doing that is actually that perhaps CUL3 has 2 modes of PD-L1 regulation, one at the transcriptional and one at the post-transcriptional level. And overall, I hope to sort of have demonstrated that with single-cell pooled CRISPR screens, we are in a unique sort of situation where we're not only connecting a single gene to the regulation of another gene, but rather a pathway to the regulation of a gene, which I think is extremely powerful. So with that, I'd like to thank all the Satija Lab members for helping me throughout this project, and also Innovation Lab members. And I'd like to thank you for your attention, and I'll take any questions.
Thank you, Effie. That was a wonderful talk. Just as a reminder, if you have questions, you can either raise your hand and I will call on you to unmute yourself, or you can type the question in the chat box and I'm gonna read it. We have a question here from Sergey Kolchenko regarding the perturbation signature calculation. How sensitive is it with respect to the number of neighbors to compute the background signature?
Thank you, Sergey. That's a great question. We actually tested a few we varied the number of K neighbors essentially in the calculation. I'll have to say that, you know, intuitively, if you go too low, like say you use 3 neighbors, chances are you are not going to, you're going to miss some of the signature you're trying to remove. For example, if you go to K's like really high, say you calculate the K 200 nearest neighbors, then you're probably grabbing neighbors that are coming from different clusters, or you know, from that have different sources of variation, and therefore you're not really doing what you were hoping to do, which is to subtract those sources of variation. So in general, I think we would recommend people can change that parameter, but anywhere between 20 to 30 neighbors should give you pretty good results.
Yeah, I'm gonna, you can unmute yourself, Sergey.
Thank you. So I was, when you were showing the t-SNP plot, and I was thinking that if you take a discrete number of neighbors, and if you have cells which are coming from like more dense areas and from sparse areas, and you're taking the same number of neighbors for the cells coming from basically these 2 different type of regions, did you consider taking kind of a neighborhood around the cell and taking all the neighbors there, kind of sort of density-based aggregation of the background signature rather than a discrete number of neighbors?
Yeah. No, we didn't do that. I don't believe so. Yeah, no, I don't think we did that. One thing we tried, which didn't change much the results, well, I guess it still depends on the number of K, but you know, we have this data set integration method in the lab. So instead of actually using the PCA space to find neighbors, we used the integration assay PCA space, which didn't seem to change at all the eventual results of Mixscape classification, which was our way of sort of comparing results across different methods.
Thank you.
I have a question myself. So to develop Mixscape, right, the algorithm assumes that there are escaping cells that will have the same like signature that the non-perturbed cells, and cells that have some sort of let's say changes in their mRNA or whatever that will be perturbed, right? But then you show also that there were some non-perturbed cells that had in-frame or frame shift mutations, right? I was wondering, would it be possible for some cells to some sort of have like a third signature that did not overlap either with the non-perturbed or with the perturbed? Yeah, I'm just curious.
Yeah, no, that's a good question. This is possible. It can be sort of an intermediate phenotype. For example, imagine like capturing a cell sort of that has just started to see some of the effects of the perturbation it has received. But you caught it like minutes after that perturbation happened instead of hours or a day. Another case where that's possible is when you start doing gene combinations. So if a cell has received more than one guide RNA and you've targeted 2 genes, now the number of possible sort of distributions you're gonna have to predict are more than just 2. And so non-targeting might not be the same as a non-perturbed class. Does that sort of answer your question?
Yeah, thank you. We have a question from David Angeles. From Yuhan. He's asking, do you observe non-perturbing cells turn to cluster with a non-targeting cell more often?
Clarify.
This is Yuan speaking. That's my question. Can you hear me?
Yeah.
So I was wondering if one of the guide RNA doesn't work, would you expect to say it's actually clustered with the non-targeting cells?
Yes. Ideally, yes. After we calculate the perturbation signature, the cell should definitely cluster with the non-targeting controls.
So if I don't have measure the score, the perturbation score, because I was wondering if they cluster with non-targeting, we could actually already separate them, right? Without calculating the perturbation score, right?
Well, the problem is that if you don't remove all your confounding sources of variation in the data set, then you won't know which cluster is specific to non-targeting control cells. You're going to have a lot of clusters with non-targeting cells, but also other types of targeted cells.
Okay, thank you. Now I see your point. Yeah, thanks.
We have another question from Athi Siviris. How adaptable are your approaches with Mixscape to Cas9 knockdowns in primary cells? It seems your work would directly address the more limited knockdowns observed with these platforms.
Yeah, we have tried, so for example, we have tried analyzing CRISPR-I data sets before with Mixscape, and it seems to be working pretty well. We can also do, this particular data set I'm analyzing comes from one cell line, meaning in theory, one cell type. So if we're looking at sort of primary cells where we have a mixture of different cell types, for example, after you classify all the different cell types you have in the data set, then you can run Mixscape in each cell type separately to sort of see the effect that each perturbation has in each cell type in your data set. Does that answer?
Yes, thank you.
Okay. Yeah, we have another question from Thomas Carroll. Ritzel, did you find that similar perturbed clusters were found after other methods for removing confounding noise, like regressing out cell cycle phase or integrating by replicate? Or did Mixscape give additional sensitivity to those methods?
So yeah, we did try to, for example, regress out cell cycle and regress out replicate ID. But these were sources of variation that we were actually, we were able to know, to have labels to know that they exist in our data set. But there were other sources of variation that were not as sort of straightforward to classify your cells as belonging to one category versus the other. Like we did have a cluster that was a stress response cluster, which we didn't know. And when we applied our perturbation signature, we end up also removing. So So the beauty about perturbation signature is that you don't necessarily need to know the classes, the sources of variation beforehand. You can just subtract out anything by finding those local neighbors. We did try integration, integrating our cells by replicate and then sort of looking or calculating the perturbation signature on those on the PCA done on the integration assay, and those seem to give similar results to our first sort of Mixscape approach. So that didn't change much.
Yeah, that makes sense. Thank you very much.
Okay.